That's the cerebral hemisphere. Okay, here's the different lobes that you guys need to know. Frontal, parietal, temporal, occipital, and the insula. All right, now there are a huge number of these particular type of neurons found within your cerebral cortex, and these collectively are referred to as the mirror neurons. The mirror neurons were discovered sometime in the mid-80s to the early 90s and stuff like that, and these particular neurons has been widely studied. And what these particular neurons do is that they basically will send electrical signal when you are experiencing events that allows you to see and mimic other activities. So let's say if you're learning how to drive, right? Let's say if you're learning how to drive a manual car, which is kind of rare nowadays, right? So let's say if somebody is shifting the gears and you're looking at it, your brain is trying to mimic that motion as well. Or if you're learning a sport, and let's say you're learning golf, and let's say you see the coach like do the swing, you're looking at that particular activities, and in your mind, your mirror neurons are mimicking that activities. So therefore, when you grab a golf club, you're trying to mimic the activities that was already produced by another person, and as you're going through it, that coach then fix your swing along the way, so therefore, as you continue to do that, you now learn a new motor activities altogether. Or if you're trying to tie a tie, you don't know what that is, you pop up Google and you look at YouTube and say, all right, how do I tie a tie? And you watch that YouTube video and you try to mimic those activities, okay? So mirror neurons are actually very, very important for the development of what we call social skills and language skills. So what do you guys think language skills is about? If you guys have ever taken a language course, you will have a writing portion, a listening portion, and an auditory portion. Does that make sense? So when you're listening to somebody talk, you're mimicking the accents. When you're writing it, you're mimicking the grammatical, like let's say structure of that new language, okay? Or when you're writing, you're recognizing the, let's say the orders of the letters that form that word. If you're learning Spanish or French or whatever the case is, right? Or if you're learning some kind of archaic language, like if you're learning Mandarin or you're learning, Sanskrit or whatever the case is, it's not based on the alphabetical stuff, it's just based on symbols. So therefore you have to also be able to kind of understand how those symbols are aligned together to form a word, right? So any of those particular activities in terms of learning social skills or language skills all highly depend upon mirror neurons. This is also one of the theory behind autism. So a child who is autistic has some degree of loss of connection of these mirror neurons, which means they can no longer apply the same mimicry. So therefore, those children that has autism has very different sort of like interpretation of social cues. They don't know how to line up. They don't know how to engage in conversation and things like that, right? And in terms of that, sometimes autism is gonna lead to a severity where they can no longer take care of their bodily needs. They can't feed themselves, they can't clothe themselves, they can't do any of that stuff. So it leads to some very, very bad motor, just basic motor skills as well. And they can't communicate effectively to tell you what they want. Children have very bad language skills, right? How do they communicate? If they're frustrated, they throw tantrums. Some adults never develop appropriate language skills. So how do they communicate their frustrations? They throw tantrums as well. Okay, so if you throw tantrums all the time, think about your mirror neurons and then find a way to develop those mirror neurons, right? Just mimic the people that you want to, that has good social skills or good language skills and stuff like that, okay? So, very important. All right, good. Questions? All right. Now, deep into the brain tissue, so all the stuff we're talking about, all these cortexes, are basically just on the periphery of the cerebral of the brain itself. Now, deep to that brain tissue, there are a collection of neurons in here as well. Now, there's a series of neurons that basically form the circuit we call the basal nuclei. You guys remember the term nucleus is a collection of neuronal cell bodies of neurons within the central nervous system. right? So the basal nuclei are a series of neurons that are located deep into the brain. And within the basal nuclei, you have different areas called the claustrum, the corpus striatum, the lentiform nucleus, and the putamen, the globus pallidus, interna and externa, and all that good stuff, which you guys don't really have to know, okay? Because that should be all neuroanatomy, which you guys should already have. Now, with the basal nuclei, it's gonna set up what we call a motor circuit. This motor circuit, with the help of this hormone or this neurotransmitter called dopamine, is gonna help regulate your motor activities. Right? So with the motor circuit, there's actually two different circuitry. One is called a direct pathway, and the other one is called the indirect pathway. You guys don't have to know all of these connections. Okay? Just know that there's two pathways that exist. And these pathways starts from the motor cortex, which is in your precentral gyrus, that primary motor cortex. Okay? The neurons there projects to other areas within what we call the basal nuclei. So all of this area that's deep into the brain itself, right? And then the direct pathway through all of these neuronal association is going to increase your motor activities. Okay? That's all it does. This is all you guys need to know. With the direct pathway of the motor circuit, if that pathway is active, you have an increase in motor activities. Okay? Whereas the indirect pathway of the motor circuit basically has almost the same association with the motor cortex. It's going to decrease your motor activities. So if you want to reach out and grab an object, you already have a plan. You have executive planning in your brain to recognize what that object is. Make sure it's not dangerous to you, because you know what's dangerous. If this is a candle, I'm not gonna grab the flame, right? I'm gonna grab the actual waxy portion of the candle. If this is a knife, I may not wanna grab it all together, okay? If this is, I don't know, whatever it is, like a burning lava or whatever it is, I'm not gonna reach out and grab it. So let's just say, I recognize the fact that this is a pen, which is a harmless object that's not gonna cause any pain or injury to my body. And I want to basically reach my hand out and grab this particular object without knocking it off the table. Okay? So therefore, through a series, and it's a combination of both direct and indirect motor circuitry that gets turned on at the same time. But it depends. Do you want to grab this object quickly, or do you want to grab this object slightly, slowly, and more controlled? Okay? So it all depends on the type of motor activities you engage in. Keep in mind, you always have both pathways being activated at the same time. This will give you a purposeful type of motor activity. Does that make sense? So purposeful means you know what your goal is in terms of producing that activities. If I'm to walk from here to that door, I already plan to do so. Does that make sense? So therefore, the only thing that I need to assess now is how quickly I need to get to that door, or how slowly I need to get to that door. Or do I need to get to that door chaotically or in an orderly fashion? Let's say if there's an alarm going off, do I just haul ass to that door and just lock it behind me and then leave everybody in here? Or do I just walk slowly and calmly to that door, open it, and allow everybody to leave before I leave, or whatever the case is, right? So everything is pre-planned, and therefore all of your motor activities have both direct and indirect pathway that's activated. The ratio of input between the direct and indirect pathway is going to modulate the intensity of that activity. That's all. If you have a higher ratio of input from your direct motor pathway, you're going to have an increase in motor activities. Basically, you can achieve that motor activity faster. Okay? But that means there's less precision when you have an increase in that direct motor pathway. If you have more of that indirect motor pathway, your motor activities can be slower, but it is a little more precise. So let's say if I'm trying to grab this pen, if I'm trying to grab it too quickly, if let's say I have a lot more direct pathway having an input, I may not grab it correctly, or I may knock it off the table, or whatever the case is, right? But if now I want to grab it, but I want to do it slower and more purposefully so that I don't basically knock it over. Let's say if I just want to grab the tip of this pen, then there's more indirect pathway input to my movement. Make sense? So that, therefore, I can have a lot more precise motor activity. Okay? So anytime you guys are like texting on your phone, that's a lot of indirect motor pathway put in there. Otherwise, you're just gonna be pounding on your keyboard. That's what you're gonna be doing. Okay? So what happens if you have only direct motor pathway or the direct pathway of your motor circuit active? What's gonna happen to your motor activities? Imagine, let's say now you wanna reach out and grab that pen, but then you have the ability to turn off the direct pathway and only turn off the indirect pathway and only turn on the direct pathway. What's gonna happen when you reach out and grab that pen? You're gonna end up slapping yourself because you have no control whatsoever on how much of this motor activity you can impose. Make sense? What happens if you turn off the direct pathway and only turn on the indirect pathway? That's your movement right there. You're not moving at all. Does that make sense? So therefore, it has to be somewhat of a ratio between the two. So you have to have equal input or some slightly different in input so that you can produce what we call a purposeful type of motor activity. Okay, so that's the difference between the direct and indirect motor pathway. Okay? Don't think of it as black and white. It's not an action potential. It's not an all or none principle. For motor activities, it's all a mixed and modulated or ratio amount of these particular input. Okay? So in order for you to reach out, do anything, say Kevin twirling his pen, that's a combination of both direct and indirect pathway. Otherwise, he won't be able to do that. If you ever pick up chopsticks and you're using chopsticks to pick up your food or whatever it is, or you're typing on your tablet, you're texting on your phone, that's a mixture of both direct and indirect pathway being inputted into your motor activity. This is how you fine-tune and modulate every single motor activities that you engage in. Okay? Because you're trying to produce some kind of goal-oriented type of movement. Okay? All right. Questions? Now, the idea of what we call cerebral lateralization is the fact that imagine your body is divided into two, right? Cerebral hemispheres also has a right and a left cerebral hemisphere, portion of your brain. Any kind of sensory information applied to the left side of your body will be relayed to the right primary somatosensory cortex. So it lateralized to the other side. Okay? Any kind of sensory information applied to your right side of the body is going to be relayed to the left primary somatosensory cortex. Okay? The left primary motor cortex is going to control the motor activities, the left primary motor cortex is going to control the motor activities of the right side of the body. Whereas the right motor cortex is going to control the motor activities of the left side of the body. So there's some degree of crossing over of these neurological fibers that controls either the motor activities or receive sensory information from the body, okay? And that's what we mean by lateralization. But that doesn't mean only the right side will detect sensation or control motor activities of the left side. I mean, it's intended to do that. However, the brain communicates drastically between the left and the right. Okay. So even if you are receiving sensory information, it is detected. Let's say the sensory information is being placed onto your left arm, it is detected by the right primary somatosensory cortex. But the other side of your brain also realizes that, also understands that is going on. Okay, so you have this bilateral kind of sort of like overall application of your brain. Okay. And the communication between the right and left side is through this portion called the corpus callosum, which just contains massive amount of tracts. And you guys remember what tracts are? Tracts are just bundles of axons that are heavily myelinated. Basically allows for these neurons to communicate back and forth between the right and the left side of the brain. Now, if you think about brain dominance basically refers to the neurons that are located on the right side versus the left side of the brain that basically may have a higher in sensitivities of activities. So therefore, the neurons that are located on the right hemisphere mainly is very good for you to detect things like visual spatial type of activities, recognizing face and objects and stuff like that, right? Recognizing or composing music, arranging blocks. If you love Legos and you like to build blocks, that's basically due to the neurons located on the right side of your brain. Reading maps, which is now a lost art. And nobody reads maps anymore, because why? Because the GPS tells you where to go, right? So you no longer have to read anything. Now, if on the left side of the brain, the neurons there are more sensitive to things like language and speech, right? Things like writing and calculation, so more technical stuff, more orderly type of structure, kind of things like music and calculation and things like that. So therefore, think about this. If you are a visual learner, that means the neurons on your right hemisphere is more sensitive to the input, all the stimulation that's being input into your brain. The right side basically is going to be a little more active than the left side. Okay? If you are a person who loves to learn through, let's say, some kind of order listing of things, right? You organize it by having an outline or whatever it is, or you structure it in a certain way, then you are more of that left brain dominance, because the neurons within the left hemisphere becomes more sensitive to the sensory input about the topic that you're trying to learn. Okay? But that doesn't mean you can't utilize the other side as well. Just because you're a visual learner doesn't mean you shouldn't learn it by making a list. Just because you're an organized learner doesn't mean you shouldn't learn it by drawing pictures. You need to do both, because you need to train your brain to seeing multiple type of input, so that if I give you a picture to interpret, or I give you a description to interpret, you should still be able to do so, right? And this also gives your brain a better chance of learning that topic, because you are now putting in different type of sensory information so that you can learn the same topic in different ways. Awesome. Okay. All right, here's the corpus callosum. There's the bridge right here that connects to the right and the left side of the brain. All right, now, in terms of language, because language is one of the most important ways for us to communicate, right? Within our social structure, within the community, and of course, internationally as well. Because language is one of the things you need to learn when you want to engage with the people around you. So language is assessed by two very important areas, and typically, these areas are located on the left side of the brain. So in the left cerebral hemisphere. One area is known as Broca's area, and the other one is known as Wernicke's area. The W is pronounced as a V, right? You can call it Wernicke if you want to, but it doesn't matter as long as you spell it correctly. Okay. So Broca's area is located in the left inferior frontal gyrus. So imagine your frontal lobe, right? The frontal lobe has a precentral gyrus, but then in front of that you have three of these horizontally placed gyri. So you have superior, middle, and inferior frontal gyrus. So around the inferior frontal gyrus is an area we call Broca's, named after Dr. Broca, the guy who basically discovered this thing, right? So Broca's area is an area that contains neurons that controls the motor aspect of speech. This area has neurons that will project to the primary motor cortex, so that the neurons in the primary motor cortex can control your tongue, your palate, your pharynx, and the muscles of your mouth and your neck and stuff like that, so that you can speak the words. You can flex your tongues, you can roll your tongues and make the sound or whatever it is that you're trying to make, right? So you can do all of that. So therefore, you can now project the words of the language you're trying to express. That's Broca's area. Now, if something happened to Broca's area, we call this Broca's aphasia. Aphasia is the inability to express language. So therefore, Broca's aphasia, in this particular case, let's say if you have something happening there, like the neurons there die or something like that, right? Then what happens is that you will have a loss of a way to broadcast the spoken word, to speak the words. So therefore, the result of that, these patients will have poor and slowly and poorly articulated speech. They're trying to get the words out, but they can't. And it's not like stuttering, it's just they can't form the words because they have lost the motor control to basically allow them to form those words. Yes.
So if I can't roll my R, is this issue?
No, no. If you can't roll your R's because you can't roll your R's. Because you don't have that high frequency of just tongue hitting the roof of your mouth or whatever the case is, right? So it has nothing to do with Broca's aphasia. But let's say if you do have a patient that you're caring for and the patient had a stroke, and now the patient can't talk, or the patient can talk, but then the speaking is very hard for the patient, and the words that comes out are very poorly articulated, right? And it's not because they have an accent or they don't know how to speak those words. They did at one point, but now because they have a loss of those neurons, they can no longer articulate that word clearly to you. So Broca's aphasia is a problem with broadcasting the words itself, okay? Now, since this is only affecting how you speak the words, it doesn't affect how you understand the language. So therefore, people with Broca's aphasia are very frustrated with their inability to express that language appropriately, okay? To speak the word, to say the word clearly, so therefore, a lot of times, like either their loved ones or whoever it is that they're talking to will be like, wait, can you say that again, or whatever, they don't understand it, right? All right, so. Now the other area called Wernicke's area. Wernicke's area is located in the left side, right next to the temporal gyrus, right next to the occipital lobe, right next to the parietal lobe. So imagine parietal lobe, occipital lobe, temporal lobe. So right at the junction of those three lobes is Wernicke's area. Now Wernicke's area is important for the comprehension of language, not just speech, because language is not only about the spoken words. Language is also about the written words, because you can read the language, you can listen to the language, and you can speak the language. Does that make sense? So when somebody speaks to you and you hear it, you can understand it. When you're reading the words on a passage, you can understand the grammatical, context of those particular words or whatever it is in that language, right? So therefore, comprehension of language is an important portion and functional portion of what we call the Wernicke's area. Now, if something happens to Wernicke's area, which means there's some damage to those neurons, we call this Wernicke's aphasia. In Wernicke's aphasia, there is nothing wrong with how you articulate the word. So you can still say the words clearly, but because there is something wrong with how you associate language and the comprehension of that language, therefore the whole context of your speech is just a bunch of words that makes no sense. You can still say the words clearly, but you cannot, those words, when you align them together, it doesn't make any kind of grammatical content. Does that make sense? So we call this a word salad. So therefore, for Broca's aphasia, this is due to the inability to broadcast the language itself. Whereas in Wernicke's aphasia, this is the result of this is the production of what we call word salad. So for example, if somebody has Wernicke's aphasia, let's say somebody had a stroke in that region of the left side of the brain, and you ask them, how are you doing today, sir? Their answer should be table, chair, floor, lamp. What? They can say the words clearly, but it doesn't make any sense at all when it comes out. Does that make sense? So there's an inability of those patients to assess the language appropriately. They don't comprehend. They can understand the words that are coming in, but when they respond to those words, it's completely off. Yeah.
So it says information about written words is sent by the occipital lobe. So does that mean like the information about like words that you hear is from the temporal?
Yeah, because this is right at the junction of those three lobes, right?
Okay, and then yes, like what would the?
So if you ask the patient to read the passage,
Yeah.
and they have Wernicke's aphasia, and you ask them, can you read this passage and relate that back to me? They'll read the passage and they'll say something completely different.
Yeah, but they'll understand like what they're reading.
They don't, no, they will not understand. That's the problem with this particular area if you damage it. Because this area is important for the comprehension of language, right? They'll read it, they may look at the word, they can still read the word. They may not understand what those words are. Maybe in their mind, they think they understand it, but it is an abnormality of the comprehension of it.
Okay.
So therefore, you lose the context,
Okay.
yeah, and the meaning of that language.
Can they still learn and understand sign language, for example?
Oh, maybe. Wernicke's aphasia. Now, these two areas communicate with each other all the time. Because in order for you to respond using Broca's, respond with language, you have to understand the language. Make sense? So you have to comprehend what it is that's being asked of you, what it is that you're reading, so that you can appropriately respond to that language in the same context. So therefore, there's a communication between the two. Here's Wernicke's, here's Broca's. Okay. There's a very intimate connection between these two centers of the brain on the left side mostly. And we say mostly because about 90% of the patient in the general population across the world, their Wernicke's and Broca's is located on the left side. Okay. There's a very small percentage of people in the world that has Wernicke's and Broca's on the right side. All right, so then you can see from Broca's area, they have a connection, a neural connection with your primary central, the prime motor cortex in the precentral gyrus. Because this area, again, controls the motor aspect of every kind of motor activities. Questions about this? All right. Okay, the next center in the brain, we call it the limbic system. Limbic system is basically what we refer to as a primitive brain, because the limbic system is basically seen in every single organism across the entirety of the organism. Well, mainly just like vertebrates, right? So with the limbic system, the series of neurons that are found within that area is allowing for you to basically assess emotions and also, very importantly, memory. Okay? So when you want to retain memory, it has to go through this system as well. Okay? Because a lot of memories that are associated with any object has some component of emotion associated with it. Right? So let's say if you're studying for this class, and you're studying, and you're sitting there, and like study, and like, I can't get in, you start crying and stuff like that. On the exam, cry, because that will trigger the memory of whatever it is that you were learning at that particular point. I mean, please don't do that, because I don't have any paper for you guys. So I'm gonna have to use your sleeve to wipe your nose and stuff like that. But you can use emotion in that sense too, to trigger the memory of whatever it is that you're trying to learn. Okay? So that's also very important. Now, the limbic system is also very important for a certain sense of motivation, right? To learn stuff and stuff like that. So it's all also in there. Therefore, if something happens to the limbic system, you basically don't have much of an emotional range. You may not have any emotional at all. Like you can sit there and you see somebody like getting killed in front of you and you're like, eh. Right? Or somebody like propose to another person, you're like, oh, okay. Right? You win $1 million, or sorry, not $1 million nowadays, $1 billion from the lottery, you're like, eh. No emotion whatsoever. Okay. All right. Now, with the limbic system, it is highly associated with olfaction. Because olfaction is a sense of smell. So therefore, the neurons that's responsible for detecting the odors in the air, the odor particles in the air, is going to directly relay their neurons to the limbic system. Okay? So therefore, the limbic system is one of the first centers of the brain that receives information about the odor within the air that you smell, the sense of smell. And because of that, you immediately associate that smell with the memory of whatever object that emanates that smell. And also, you relate that smell with the object and an emotional reaction to that smell. Okay? So think about any smell that you can smell. If I put a rose in front of you and you guys smell that rose, most of you guys be like, oh, roses, beautiful flower, Valentine's Day, love, whatever. Right? If I smell a rose, I start to tear up and like I cringe in pain because maybe the memory I associated with the rose was once upon a time I gave somebody a rose and they just slapped me across the face and all the thorns like cut me and stuff like that. So now it's a very negative memory. Right? So therefore, it all depends on the memory you associate with the smell and the object that emanates that smell. Make sense? Right? So that's why you smell an apple, you think an apple. Some people smells an apple and think grandma. Why? Because Maybe in their youth, every single time they go to grandma's house, grandma's always making apple pies. So therefore they associate that smell, which is coming from the apple, with the apple pies that's made by grandma, right? So. All right, here's a bunch of emotions that is regulated by the limbic system. You guys don't really have to know this, you can read it, right? So you can think like these areas is more for aggression, fear, hunger, satiety, sex drive. That's a whole limbic system altogether, right? So therefore it's a very, very powerful drive, emotional drive and stuff like that. And that's why you have a lot of crimes being committed because of pseudo romantic type of notions, which is just maybe just sexually oriented. Anyway, we don't need to get into that. That's a psychiatry class altogether. Okay. Now, there's two areas in the limbic system that is allowing you to assess emotion and associate that emotion with memory. And the area that allow you to associate that emotion is called the amygdala. And the area that allows you to retain the memory is called the hippocampus. And both of those areas are part of the overall limbic system. Okay? So keep in mind, remember I told you guys at one point, right, the different stress levels that you may experience when you're learning things. When you are learning something, you are stressed, and that stress releases cortisol, which is your primary stress hormone. That cortisol is going to act on the amygdala and the hippocampus, mainly, and allows that area to retain the information that you're learning. And you will retain it and you will convert it into somewhat of stored memory. So you will have all the information locked inside your brain. Okay? However, when you're sitting for the exam and you're a little more stressed out during that time, more cortisol is released. But now because you have a systemic stress, that cortisol stimulates the release of glucose, which then gets divided throughout the different tissues of the body. And that cortisol actually will now inhibit the neurons within the hippocampus and the amygdala to bring out that memory. So during that high stress environment, you actually have a decrease in memory recall. Okay? And so think about it this way, it's very simple. Let's say if you learn to drive, right? You know how to drive, everybody knows how to drive. There's a steering wheel, there's a pedal on the right for acceleration, and a big pedal on the left to brake. If you drive a manual car, there's a third pedal for you to kind of engage and disengage your gears and stuff like that. So let's say you're driving an automatic car, right? So you're sitting there, you're like, oh, green light, step on the accelerator and you go. Change lane, doop, 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 and then you change lane, you look over and make sure you change lane. Okay, so everybody knows how to drive, right? However, let's say if you're driving and all of a sudden somebody cuts you off immediately into your lane and you're like, oh my God. So you have these extreme movements, which means at that point, for some reason, you are no longer able to recall the appropriate driving maneuvers. Because now you're in a very high stress situation, right? So how do you overcome that? Well, maybe you have to engage in some kind of defensive driving school. Or maybe you can be a better defensive driver, let's say, be more aware of your situation, so you know, oh, this person's about to cut you off. You can pre-plan, sort of like your escape route, right? Pre-plan what you're gonna do in response to that. Or let's say if somebody attacks you and you freeze up, you don't know what to do. Well, why is that? You know how to move your arms and legs, you know how to turn and then just get your ass out of the way and run, but why do you freeze up? Like you get into a confrontation, so somebody throws a punch and you're like, oh, I see a fist coming at me. I should do something about it. Too late, right? Why is it that you freeze up during that time? Well, because under stress or situation, recalling of activities is inhibited. And recalling of movement is also an activity. So imagine that's happening in terms of, let's say, just psychological stuff. If you're trying to recall the information that you learned, it's also inhibited. So you know it, you just can't recall it. That's the worst thing. How do you overcome it? Repetition, repetition, repetition. Why is it that you think these mixed martial artists or anybody engaged in some kind of, let's say, martial arts or firearm trainings or defensive driving trainings or, you know, race car drivers and stuff like that, why do they have super fast reflexes and they can remain calm even though they're going at 200 miles per hour and somebody's trying to cut them off? Why is it that they can maneuver in a very controlled situation so that they don't get into some kind of, let's say, accident? Because they have repeated training. So if you can train yourself so that you can remain calm, which means repetition, 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 then you can recall things a lot easier. Right? Like I told you guys, how many of you guys walk out that door and immediately said, ah, fuck, what did I choose B? It should have been C, right? You know it, just can't recall it. So stop doing that to yourselves. Okay? Because it's not the questions that's tricking you, it's your brain that's tricking you. Okay? Keep that in mind, that's how you do it. All right, anyhow. And this you guys don't have to know. This is just basically, you know, other emotion and memory and stuff like that that's involved with the limbic system and all that good stuff. Okay, now you can see if you kind of scroll through this, there's a lot of different types of memory that you can retain in your brain, right? It doesn't matter which type of memory, but when you are exposed to a topic, any particular topic that you're trying to learn for the first few times, the memory that you retain is what we call short-term memory or working memory. Basically, it's the memory, the repetition that you're trying to go through so that you can fully understand the process of what's happening, okay? So as you're learning it, all of this memory that you're trying to store is referred to as short-term memory or working memory. The trick is to turn this working memory into what we call long-term memory. Okay, sort of like the long-term memory is our memory of how you recall the faces of your loved ones, how you can immediately recall your address, you can immediately recall your birth date, and maybe your social security numbers, and you know, or maybe not bank account, and nobody remembers like all 10 digits of your bank account, right? But you can recall your phone number very easily. Why is that? Because you see it all the time, you repeat it all the time, and there's no stress in recalling it. But I assure you, you will have stress if somebody force you to do it. Okay? If you're sitting there and you're like trying to put in your PIN number or whatever, and somebody has a gun to your head, you're like, oh, fuck, there's so many numbers. It's only 9 numbers, 10 numbers. It's just in whatever sequence you remember it to be, right? But why is it that you can't recall that? Well, because you're under duress. So train yourself to basically convert what we call working memory into long-term memory, okay? And long-term memory requires strong association between the neurons that are relaying that information back and forth. Now, that strong association also is, you must also change the anatomy of that neuron to a certain extent, so that they can basically transmit neurotransmitter more effectively and efficiently between the two neurons. That's all it is. So let's say you have 2 neurons in a synapse, right? A connection between 2 neurons. If you stimulate this neuron, well, how quickly can they release neurotransmitters? And if they release neurotransmitter, how quickly does the postsynaptic neuron reach the threshold to produce an action potential? Because as soon as this postsynaptic neuron produces an action potential, it can relay the information very quickly so that you can recall it. Makes sense, right? Okay. So therefore, with long-term memory being converted into, short-term memory being converted to long-term memory, we call this memory consolidation. Okay? And with memory consolidation, you have to now actually impose anatomical changes to the neurons itself. And it is also enhanced with adequate amount of sleep. And I'll show you guys how to do that. Okay, as far as long-term memory, there's 2 different types of long-term memory, okay? There's what we call non-declarative memory, which is memory of how to do things, and this is referred to as implicit memory. How to do things like how to tie your shoelace, how to tie a tie, how to comb your hair, how to brush your teeth, things like that, right? How to write, how to drive, okay? The other type of long-term memory is called declarative memory. Declarative memory is also referred to as explicit memory. This is basically how to recall facts and dates and things like that, okay? So for declarative memory, there's two different types. There's semantic, which is basically recalling facts, and there's episodic, which is essentially recalling events, birthdays, anniversaries, like historical dates and stuff like that. And those historical dates can also be facts as well, because those are what you learn, right? So birthday is something that you can experience, so therefore it falls under this episodic type of events, okay? All right, so two different types, right? Non-declarative versus declarative memory. Both of those are long-term memories. So how do we do that? Well, let me draw it out for you. So this is a neurological phenomenon where you have to now change the synapse. You have to make the synapse between the pre and the postsynaptic neuron much stronger. Okay, so imagine, if you will, you have a presynaptic axon terminal. Here, let's say you have this dendrite over here. So presynaptic neuron. Let's say we'll call this number 1. Okay, let's say you have multiple, right? So you have another one here. This is presynaptic neuron 2. Okay, this is a postsynaptic neuron here. Okay. Now, imagine if these neurons are forming a circuit that allows you to retain memory of something that you're learning and allow you to also recall memory of something that you've just learned. Okay? So for this particular neuron, it has a bunch of vesicles. Same thing here, right? The axon terminals of these neurons have vesicles. You guys remember the lecture about synapses and stuff like that? Okay. Within this vesicle, there are a bunch of neurotransmitters. Imagine these neurotransmitters are a neurotransmitter we call glutamate. What are glutamates? Talked about this last time. Excitatory neurotransmitter. It is the primary excitatory neurotransmitter. Okay? And you have these neurotransmitters here inside this vesicle of this other neuron, and these guys are GABA. What are GABA?
Inhibitory.
Major inhibitory neurotransmitter in the central nervous system. Okay? All right. So, imagine you have an action potential traveling down this axon all the way to the axon terminal. It triggers the activities of this voltage-gated calcium channel. Opens up, allows calcium to move into the cell. Calcium is going to combine with a protein that forms a complex that will activate the snare protein. The snare protein is where this vesicle is docked on. So therefore, once the snare protein is activated, it allows for fusion of this vesicle to this membrane. You guys recall this? Please do. Because if you don't, that's a lot of points that you're gonna miss. I guarantee you, this test is the hardest one in this class, okay? Most people will fail the second exam because most people will find it too hard to really even study it. Don't do that. Nothing is hard. You just refuse to study it. Don't refuse to study it. Tell your brain, it is not that hard. Once you say it's hard, you've already given up. Okay, so do not tell yourself it's hard to know. There's nothing hard about this stuff. It's going on inside your head every single minute or minute second. All right, so you have a release of this glutamate, okay? Once this glutamate is released, it will bind to some receptors on here. So imagine there's two different types of receptors. These are both specific for glutamate. And these are ligand-gated receptors specific for glutamate. Remember I told you guys, there's 3 receptors specific for glutamate. 2 of them are ligand-gated and one of them is a G protein coupled receptor. The two that is ligand gated, that's specific for glutamate, is either an NMDA receptor or an AMPA receptor. Okay, so here's an NMDA receptor. And here's an AMPA receptor. Okay, that R just basically stands for receptor. Now on this side, let's say if you want to release this GABA, again, it's the same concept. Okay, action potential travels down, stimulates this voltage-gated calcium channel, allows the calcium to flow into the cell. Calcium is going to combine with a protein, activate the snare protein, fusion occurs, and when you fuse, you allow for the release of this GABA. On the postsynaptic surface of this neuron, there are also ligand-gated ion channels specific for GABA. What do we call those? We will have this conversation, You guys had a little more than 24 hours to learn this stuff, right? If you can study this, you're already behind. So there's no reason for you guys not to study this stuff once you're already exposed to it. Like I said, you need to study, you need to take at least 2 or 3 hours every single day to learn this. Otherwise, you're gonna be behind. What is it? It's a GABA receptor. And what kind of receptor is a GABA receptor? Ligand gated or G protein coupled?
Ligand
It's
gated.
ligand gated, yeah. Good. Okay, now I'm gonna map out the electrical activities that's being produced in this postsynaptic neuron, okay? So you guys remember the action potentials, right? So imagine here's negative 70 millivolts. Okay, this is all in postsynaptic. Okay, so in the postsynaptic neuron. Okay, so here's the resting membrane potential. Okay, imagine here's going to be your graded potential. Which means this is threshold, and once threshold is hit, then you have depolarization. Okay, this is depol. We're just gonna focus on this graded potential right here. What's threshold voltage? What's the quantifying measurement of threshold voltage?
Negative 55 millivolts.
Negative 55 millivolts, excellent. Okay, good. All right, so imagine within this postsynaptic neurons, all of this is happening, right? Now, if you have these glutamate binding to the AMPA receptor, it's gonna open up. And the AMPA receptor is specific for sodium ions. So once it opens, sodium ion is gonna move into the cell. Does that make sense? Because there's more sodium outside versus inside of the cell. So sodium ions diffuses in, and as sodium diffuses in, what does it produce? What electrical membrane potential change will sodium influx produce?
Excitatory, EPSP.
Okay, and EPSP is a depolarization, correct?
Yeah.
Okay, good. So it depolarizes this area. So depolarization. So yes, EPSP. Because remember, all of these depolarization here are tiny electrical signals. Okay, that's happening at the dendrites and cell bodies of this postsynaptic neuron. Good. Now let's say if you have GABA binding right? And as GABA binds, this GABA receptor opens up and it is specific for chloride ions. And chloride enters the cell. because there's more chloride outside versus inside. If chloride enters the cell, what's gonna happen to this part of this neuron?
Repolarize.
It hasn't depolarized yet.
Oh. Inactive.
Hyperpolarized, good. And remember, as it hyperpolarizes, it produces inhibitory signals. So therefore produces IPSPs. Make sense? That's just the normal activities of every single neuron that exists within your body, including the neurons within the central nervous system. Okay? All right, so now, this glutamate, as it binds to AMPA receptor, it will cause a depolarization or EPSP effect. The EPSP or the depolarization is going to trigger the opening of the NMDA receptor. Now keep in mind that glutamate is gonna bind equally to both of these receptors. Because both of them are specific for glutamate. However, the NMDA receptors on the cytosolic side has a magnesium that basically block the entrance of the ions moving into the cell through the NMDA receptor. And in order for that magnesium to be kicked off, you need to allow for sodium to move into the cell through the AMPA receptors to depolarize this neuron. That depolarization causes the magnesium to be displaced. Make sense? Okay. Now, once the magnesium is moved out of the way, then this NMDA receptor will open and it is specific for calcium ion. So if calcium ions move into the cell, what's gonna happen to this inside the cell? Depolarizes also. Good. So therefore also produce EPSPs. So if you take all the EPSPs and IPSPs and you add them all together, you get the greater potential, right? Okay, so remember, imagine this is what's going on in this postsynaptic neuron. All right, now this depolarization effect due to the influx of calcium, that calcium will then stimulate. So imagine inside this neuron, there's a bunch of vesicles, and those vesicles has AMPA receptors integrated onto that vesicle, just sitting there waiting. Nothing's happening to these guys, so you have a bunch of AMPA receptors here. This calcium is going to stimulate this vesicle, and the vesicle is now going to move toward the cell membrane. And as the vesicle moves toward the cell membrane, it's gonna fuse with that cell membrane, and it's gonna place those AMPA receptors onto the surface of that cell. Make sense? So now you have more AMPA receptor. Does that make sense? So if you have more AMPA receptor, what's gonna happen to the glutamate released by this presynaptic neuron? It will bind to more of those AMPA receptors. So now that glutamate can basically bind to more of these guys. And if the glutamate can bind to more of those AMPA receptors, and these guys open, the sodium ions can move into the cell much faster. If more sodium ion starts to move into the cell, what do you generate now? So you have increase in depolarization, right? So therefore you have a higher amount of EPSPs. Does that make sense? If you have more EPSPs compared to IPSPs, what will your greater potential look like? It's gonna take less time to reach threshold. So now, as you're learning any new things, this is happening. As you're learning something new, let's say if you're working through this particular memory for the very first time or the second or third time, doesn't really matter. Now you have calcium entering this particular cell, this neuron, and activating a bunch of these vesicles to place more AMPA receptors on the surface of this neuron. Which means the next time this presynaptic neuron releases the glutamate, glutamate can bind to more AMPA receptors. And as it binds to more AMPA receptor, it increases the depolarization or increases the amount of EPSPs generated in this postsynaptic neuron. So what happens to your grade potential is that it starts to look like this now. Okay? Yep.
So would this be considered a positive feedback loop?
In a sense, not necessarily, but it's just a reaction to the repeated exposure to the same material that you're trying to learn. Okay? But this is not enough. This is not enough to convert short-term memory into long-term memory. Yes, question?
I was just wondering if the NMDA receptor, like all of them got activated on the first round of AMPA,
Mhm.
or is it like continuously as more?
Well, it gets activated the first time, let's just say, but then let's say the second time, because there's more AMPA receptor, it gets activated faster. So as it gets activated faster, more calcium is flowing into the cell now, okay? So let's say if you stop learning the material at this point, what's gonna happen is that the cell, this neuron, is gonna take away these AMPA receptors. So that's the reason why you can't recall the information as well if you don't repeat the same information repeatedly. If you don't continue to engage in this particular topic, you're gonna forget about it. Right? How many of you guys have already forgotten about anatomy and you just took it last semester? Just think about that, okay? So therefore, if you don't constantly engage yourself in whatever material or topic that you're trying to learn, you will start to lose some degree of memory recall. It doesn't mean you lose it altogether, it's there. You just have to now refresh your memory so it can strengthen the recall, okay? So anyhow, so now, you think about this, now at the next time this neuron releases glutamate, it will bind to more AMPA receptors and therefore increases the amount of EPSPs. That basically increases the amount of NMDA receptors that will open. So let's say you have more NMDA receptors being triggered by this increase in depolarization. So you have more calcium moving into the cell altogether, right? This increase in calcium influx here will now allow the calcium to move into the nucleus of this cell. So again, this is through another sort of like series of repetition of whatever it is that you're trying to learn. The calcium influx is going to activate, so calcium here is going to activate what we call CREB. Okay. And this CREB is sort of like a transcription factor, and this CREB is going to activate your gene. So increase transcription of this gene. Okay, so increase the transcription of whatever this gene is. I forgot to mention the fact that, I forgot to mention that calcium as it moves in, it activates an enzyme called CAMK2. Okay, so calcium calmodulin ATP, whatever it is. So that CAMK2, protein kinase. So here, calcium, we'll add this to this. Calcium's going to activate CAMK2, and CAMK2 is the one that activates this vesicle and brings it to the membrane. Okay? And if you continue to learn whatever it is that you're learning, the next time there's more calcium entering your cell, and the calcium will move into this nucleus, activate this CREB, which is a transcription factor, which will increase your transcription of this gene. And this gene codes for AMPA receptors. So it's an AMPA receptor gene, okay? Which means you stimulate this postsynaptic neuron to make more AMPA receptors. Does that make sense? Okay. There's also another gene, and the other gene is basically a structural protein. So a gene that codes for structural proteins like cytoskeleton, stuff like that. So you also increase the transcription, translation, or the production of structural proteins inside this cell. Okay. And the reason for that is if you're making more AMPA receptors, so let's say you have a bunch of these vesicles here, because you just generated a bunch of AMPA receptors, right? So therefore, there's a bunch of vesicles transporting these AMPA receptors. Okay. But if you don't have enough space on the membrane of these neurons, where are you gonna put these AMPA receptors? Make sense? So these structural proteins is gonna change the anatomy of this membrane. And imagine if this is the dendrite of this neuron. So what happens is that these structural proteins is gonna cause these membrane to basically form these little convolutions, so to speak. Right? So the membrane of this dendrite starts to fold upward. And as it folds upward, this is due to the structural protein causing the anatomical change of your dendrites. We call this the dendritic spines. So in this repetition of information that you're trying to learn, once the structural protein has been expressed more, you now form what we call dendritic spines. And the dendritic spine essentially just increase the surface area of this particular dendrite so that you can insert more AMPA receptors. Make sense? So now you have more surface area for AMPA receptors to be placed on here. Okay. So far so good? Everybody still with me? Okay. Now you have all of these AMPA receptors, but you may not have adequate amount of glutamate. So how do you stimulate this neuron to release more glutamate so that it can bind to all these extra AMPA receptors? Well, there's a neurotransmitter, and that neurotransmitter is a gas we call nitric oxide. Okay? So therefore, this neuron is going to produce that gas. So this neuron starts to produce nitric oxide. As you continue to study, right, or continue to learn this thing, that nitric oxide, being a gas, is going to retrograde into this presynaptic neuron. And it's going to stimulate this presynaptic neuron to increase the release of glutamate. So, stimulate, increase release of neurotransmitter.
Wait, sorry, so for the dendritic spines, you're the result of the increased structural protein?
Yeah, because now you have more structural protein. Those cytoskeletal proteins will now push onto the membrane of this dendrite to form these dendritic spines. All right, so which means now you have an increase in surface area, so you can place more of those AMPA receptors on there.
And which processes does the postsynaptic cell just decided to release the nitric oxide?
Why does it?
Which part?
Oh, so therefore, once this extra AMPA receptor is now placed on there, this postsynaptic neuron knows that you have an excess amount of AMPA receptors, but you may not have adequate amount of glutamate. So therefore, it starts to release the nitric oxide so that it can stimulate this presynaptic neuron to release more of its glutamate, right? You may ask, well, why doesn't this nitric oxide stimulate an increased release of GABA? It can, but typically this neuron knows where these particular activities are, so therefore when they release the nitric oxide, they're gonna release it to the area that contains all of these neurons that secretes the glutamate, more so than to the areas that have neurons that secretes the GABA. Make sense? So it can actually kind of figure out where those regions are and release those neurotransmitters appropriately. So let's say around the area where you have neurons that release glutamate, they're gonna release nitric oxide. The nitric oxide will stimulate an increase in release of this glutamate. So therefore, the next time you're exposed to whatever topic that you're learning, this presynaptic neuron will now release more glutamate. So increase in amount of glutamate that's released, right? Now this increase in glutamate that's released is gonna bind to all of this extra or increase in AMPA receptors expressed on this surface. If you increase the amount of binding of glutamate to all these AMPA receptors, what's gonna happen to the influx of sodium ion? Much higher than before, right? So imagine the amount of sodium ions now moving into this cell is very rapidly moving through all of these extra AMPA receptors. So the amount of EPSP, so the depolarization here is much higher. Okay, so you are generating now a huge amount of EPSPs. What is this gonna look like now?
It's going to take shorter time to go
It's
up.
gonna get steeper and steeper, right? So now, it's getting to threshold much faster. Oh, but that's not enough. Okay, because you still have a bunch of these IPSPs. So the last step to complete what we call long-term potentiation, to convert short-term memory into long-term memory, is to get rid of all of these IPSPs. Okay? And in order for these IPSPs to basically be inhibited, this increase in depolarization here is going to essentially stimulate this cell to release a neurotransmitter we call endocannabinoids. Okay. That endocannabinoids is a retrograde neurotransmitter which then will inhibit the release of GABA. So this one inhibits the release of GABA. When you inhibit the release of GABA, you basically, this neurotransmitter is gonna stop this from happening, so prevent the fusion. If it prevents the fusion, it's gonna prevent the exocytosis of GABA. If it prevents the exocytosis of GABA, it prevents the binding of GABA, and therefore preventing this ion channel from allowing chloride to enter, and therefore preventing the production of IPSPs. Okay? What do we call this process? We call it, so the release of endocannabinoid. The release of endocannabinoid is due to an increase in depolarization. So this is known as depolarization induced suppression of inhibition. Because the depolarization, the excess amount of depolarization stimulates the release of endocannabinoids, which suppresses the release of GABA, which is an inhibitory neurotransmitter, and therefore you are now suppressing all of this IPSP production. Make sense? So we call this, and it's abbreviated as DSI. Now, what's gonna happen to this graded potential? Yeah, it's a straight shot, basically. Okay? So when this happens, what's gonna happen to this postsynaptic neuron as soon as glutamate is released? It's like almost immediately, this postsynaptic neuron will reach threshold and boom, produce an action potential. There's no delay because the graded potential that is being produced in this postsynaptic neuron is no longer a factor. Which means as soon as this postsynaptic neuron binds to that glutamate, it will depolarize so rapidly to threshold that it will produce an action potential almost immediately. So what you've essentially done is to strengthen the synapse between the pre and the postsynaptic neuron. Which means as soon as the presynaptic neuron releases glutamate, the postsynaptic neuron will produce an action potential. There's virtually no delay in that graded potential whatsoever. Because there's no longer any IPSPs. Right?
For the postsynaptic axon, or was it depolarization induced suppression of inhibition?
Mm-hmm.
For that one, would it like dies because because there's no use of the GABA? Or
No, it
is
doesn't
it
matter.
gonna be producing the glutamate instead?
For this neuron,
For the, yeah,
what does it release at the end?
no, I'm talking about the other ones.
Oh, this neuron?
Yeah.
Oh, what happens to this presynaptic neuron?
Mhm.
It may, basically, it's still there, right? It may not be as active, and yeah, oftentimes it can be taken away, the synapse gets taken away. But then if you don't continue to sort of like expose yourself to whatever topic that you're learning, these synapses can reestablish itself. Because as soon as this depolarization effect decreases, because if, let's say, if you learn this stuff and then you forget about it for the next, I don't know, 3 months, because you're gonna go on vacation or whatever the case is, right? And then you're applying to school and then you're not like brushing up on this. So if the first thing that will happen is that the amount of nitric oxide and the amount of endocannabinoids is gonna decrease. Right? So if the nitric oxide decreases, this glutamate will decrease in its secretion, or this neuron will decrease in glutamate secretion, which means this depolarization is not as strong anymore, which means the endocannabinoids may not be released adequately, which means that inhibition is gonna now reestablish itself. Because this neuron can now release GABA once again. So therefore, this rate of potential now becomes this, and eventually back to this again. This is what happens 5 years, or not even 5 years, 2 weeks down the road. If you don't learn this stuff well, 2 weeks later, you're not gonna be able to recall it. But surprisingly, you have it in your memory. You just didn't, or you haven't trained yourself to recall it appropriately yet.
But then, is it true that like let's say like you form that connection and then like you over time like slowly lose it, but then like the second time that you try to form that connection again, it's like easier. Or, like,
It's easier, absolutely.
yeah.
Because you already have some degree of, let's say, familiarity between these two neurons, right? So it's kind of pre-trained the neurons already.
Is it because there is some of the, I guess, like the AMPA stays?
Yeah, they're still on. Because that particular anatomical change can be semi-permanent. Right? It's harder to take away the receptors than it is to stop the release of glutamate. Right? So, yes.
Is there a capacity to long-term memory, or is this like an infinite process?
Well, there is definitely a capacity. The limitation is due to the anatomical and the chemical release facilitated by the neurons. Yeah, they do reach a max though. They do reach sort of like the max capability, right? Let's say if the amount of neurons that release glutamate is limited, then you can only reach that max capacity of the synapse. If you don't adequately inhibit all of these GABA releasing this neuron here, then you still have some degree of IPSPs. Does that make sense? So therefore, there has to be some neurological sort of like activities that can regulate all this stuff, right? It's very dynamic all the time. It's never static. It's not like, oh, once you inhibit it, it's permanent, right? It's not like once you increase the release here, it's always gonna increase the release. Yes, it will have an increase in sensitivity of the activities, which means the next time, it will release a lot more glutamate for you every single time. But if you stop learning it for a long time, or you stop exposing yourself to this material for a long time, then you stop recalling it in that immediate sense, because now you have to have some time for this great potential to reach that threshold, right? But if you continue to have to expose yourself to this, so this is what happens when you guys learn physiology for the first time. This is what happens when I ask you to recall your phone number. This is what happens when I ask you to recall the faces of your loved ones. Make sense? If you just get into a relationship, this may be what is going on when I'm asking you to recall your anniversary date, or even to recall the face of your new partner. Like, wait, is that her? Is that her? Is that him? Is that who? I don't know. You know, they all look the same. Well, don't do that. You're in trouble. Make sense, right? So that's long-term potentiation. Okay, this is what you guys are trying to achieve when you study. If you don't study, you will not achieve this. Yes.
Is that why it's so helpful that the neuron don't duplicate?
In a sense, because first of all, there's not enough space for you to continually regenerate neurons, right? Then you just have too much, and then the information never goes anywhere. So that's also another way to regulate the amount of neurons as well, because the region in the brain is limited. That's all. But you can definitely lose them. All right, good. Questions about this? Okay, so please know the steps of long-term potentiation. Okay, know how you get from here to here, and from here to here, and from here to there. This is the final stage. And so therefore, this step right here, that we call depolarization-induced suppression of inhibition, is actually an activity we call long-term depression. You are depressing the activities of this presynaptic neuron so that you can basically strengthen the synapse between these two neurons. Does that make sense? So in memory consolidation, when you're converting short-term into long-term memory, it's a series of long-term potentiation and long-term depression happening together. Okay? Because so long-term potentiation means strengthening of a synapse. You're strengthening these two synapses, or that synapse, and then long-term depression is to weaken the synapse. So you're weakening this synapse right here, so that you can pass information very quickly between these two neurons. This is what the dendritic spines look like. You see all of this sort of like convolution, these little branches that pops off on the membrane of the dendrite. This increases the surface area, so you can insert more AMPA receptors. And then that's the rest of that. Good question? Yep.
So just to be clear, like when the calcium ion comes in, like it binds with MK.
Yeah, well, they'll bind and activate the CAMK2, activate
Okay.
this protein kinase.
Okay, and then that's the protein kinase that I guess like releases the AMPA, and that's also
Yeah.
the thing that goes into the nucleus.
Correct. So the CAMK2 will activate this vesicle initially, and then if you have repeated exposure, then more NMDA receptors will open, and they'll open easier, right? So therefore, that's gonna be more calcium influx the next time. And that increase in calcium influx is again going to activate the CAMK2, and this time the CAMK2 moves into the nucleus to activate the CREB. So if you don't have repeated exposure, that CAMK2 will not translocate into the nucleus. And if you cannot allow this thing to move into the nucleus, you will not increase the expression of these genes. Make sense?
Yeah.
So you can't increase the expression of these genes, you can't make any more AMPA receptors, you can't start to change the dendrites. Right? And that's why you can't retain that memory. You can't change the grade potential duration. So you have to do all that. All right, let's go to the lab. We'll talk about sleep next time.